I'm Martha Bulyk, and it's my pleasure to introduce you to the motivation for our developing PADIT-seq technology. The study was funded primarily by grants from NHGRI, as well as a fellowship from the American Heart Association. My group's research is focused on sequence-specific transcription factors and cis-regulatory elements, which are the regions of the genome that control gene expression. The interactions between these factors and elements play a primary role in controlling gene expression programs throughout the life of an organism and also in various synthetic biology applications. There's about 1,600 human transcription factors, and understanding what controls their binding to their DNA target sites is important for understanding how they regulate a wide range of cellular processes. TF genomic occupancy profiles in a chosen cell type or state can be determined by ChIP-seq, cut and run, and other technologies. However, the identified bound peaks can reflect direct or indirect binding to DNA. There's limited availability of suitable TF-specific antibodies. There can be low signal over background, and the assays themselves are low throughput. Furthermore, the mechanisms of TF-DNA binding can be difficult to elucidate from the resulting occupancy data alone. Numerous different types of highly parallel reporter assays allow screens of very large collections of DNA sequences for their effects on reporter expression in a particular cell type. However, the interacting TFs are only inferred and not directly measured. Switching to biochemical assays, my lab previously developed protein binding microarray known as PBM technology. In the widely used version called universal PBMs, TF binding to all possible 8 base pair sequences is assayed in a multiplex format. ChIP-seq and high-throughput or HT-SELEX experiments assay a protein for binding to sequences enriched from a highly degenerate DNA sequence library. Other biochemical approaches use microfluidics devices to assay TF-DNA binding, such as in SMILE-seq and STAMP, in which TF binding to a pool of random DNA or to a few particular DNA sequences is assayed. Importantly, both high and low affinity TF-DNA binding sites play important roles in gene regulation. A number of papers over more than 20 years by my group and others have revealed various important roles of low affinity TF-DNA binding sites in gene regulation, in particular their sensitivity to TF levels for precise gene expression patterns during development. Some of these insights were enabled by data generated using some of these high-throughput technologies, including PBMs and ChIP-seq. However, it has been challenging to accurately identify low-affinity TF-DNA binding sites in a comprehensive manner. Therefore, we had a goal to develop a highly parallel, many-by-all technology that would allow us to assay TF proteins for binding to all potential DNA binding sites, here, all 10 base pair sequences, in a manner to obtain quantitative DNA binding data. My postdoc Shubham Khetan will now tell you more about PADIT-seq and the insights that the data revealed.
Hi, I'm Shubham Khetan. I'm a postdoc in Dr. Martha Bulyk's lab. And as she mentioned earlier, the problem is that current high-throughput approaches miss a lot of the lower affinity DNA binding sites. And so we need a more sensitive, high-throughput technology to measure transcription factor DNA interactions. So in order to solve this problem, we developed PADIT-seq, which stands for protein affinity to DNA by in vitro transcription and RNA sequencing. Here's how it works. Your DNA binding domain of interest is cloned downstream of a T7 promoter, and the sequence is embedded in an in vitro transcription and translation reaction mixture. And after the DNA binding domain is produced, it binds to candidate transcription factor binding sites. If it binds, it's going to recruit a T7 RNA polymerase by a protein-protein interaction and lead to increased reporter gene expression. Each reporter gene has barcodes which correspond to a particular binding site that was occupied by the DNA binding domain. In our assay, we profiled all possible 10 base pair DNA sequences as candidate transcription factor binding sites. That's a little bit more than 1 million different DNA sequences. After the in vitro transcription and translation reaction is complete, I purify the reported genes and sequence the barcodes. The number of times I see a barcode tells me how strongly the transcription factor binding site was occupied by the DBD. In order to benchmark PADIT-seq, I compared it to PBM E-scores, which is shown on the x-axis here. PBM E-scores rank order the DNA binding preference of a transcription factor to all possible 8 base pair DNA sequences. In this case, I'm showing you the data for HOXD13, which is a homeodomain. On the y-axis, I'm showing you the PADIT-seq data, and the points are colored red or black based on whether HOXD13 significantly increased reporter gene expression or not when an 8 base pair DNA sequence was occupied. So for HOXD13, we detected around 1,700 8 base pair DNA sequences that significantly increased reporter gene expression. And for EGR1, we detected around 509 base pair DNA sequences that significantly increased reporter gene expression. The concordance between PADIT-seq and PBMs is very high. The area under the receiver operating characteristic is around 0.985 for both transcription factors. However, PBM E-scores do not do a very good job of differentiating the red versus the black points very well because you need different thresholds to define them using PBM e-scores alone. And now if we overlay the HT-SELEX data on top of this, we can clearly see that HT-SELEX is biased for detecting mostly the highest affinity interactions, and it misses the lower affinity interactions that we now detect with PADIT-seq. So now that we've identified hundreds of novel lower affinity DNA binding sites, the question arises, where are they in the genome and what do they do? In order to answer this question, I performed a very simple analysis. So here I'm showing you an example ChIP-seq peak for HOXD13. I performed a sliding window analysis. So here every dot represents an 8 base pair DNA sequence, and I'm moving in 1 base pair steps and plotting the corresponding PADIT-seq activity on the y-axis. The surprising result is that instead of an individual isolated binding site, we observe 6 consecutive overlapping DNA binding sites for HOXD13 in this ChIP-seq peak. This is a surprising result because all 6 of these binding sites were profiled independently, but they occur consecutively in the genome. We see the same thing for EGR1. So we see 4 consecutive overlapping binding sites instead of a single isolated binding site. And these are not isolated examples. In general, ChIP-seq peaks contain significantly more consecutive overlapping binding sites than you would expect by chance, as shown in the histogram here. If you zoom into ChIP-seq peaks that contain 6 overlapping binding sites for HOXD13 or 4 overlapping binding sites for EGR1, there is a very interesting pattern where nucleotides flanking the high affinity binding sites create overlapping lower affinity sites. So the picture that emerges is that instead of the traditional way of thinking of genomic occupancy that does not take into account lower affinity sites, what our data suggests is that TF genomic occupancy is determined by multiple overlapping binding sites instead of individual isolated binding sites. I encourage you to read the paper for more details on the evidence that we provide in support of the model, but I would like to provide a vignette of an interesting puzzle that our model solves. It's been known that nucleotides flanking motif matches in the genome alter transcription factor occupancy, and this is a result that motif-based models cannot explain. So here's an example of a SNP that is quite far away from the motif for HOXD13. According to the motif-based model, it should not impact HOXD13 binding. However, in our custom PBM data, you can clearly see that the reference allele is occupied by HOXD13 to a higher level than the alternate allele. When we tile PADIT-seq data on both the reference allele and the alternate allele, you can clearly see that the reference allele has 1 additional overlapping binding site, and it explains why it is occupied by HOXD13 to a higher degree in our custom PBM experiments. Now, this is not an isolated example. In general, we see that PADIT-seq outperforms motif-based approaches in predicting the effects of non-coding variants. So in conclusion, PADIT-seq detects hundreds of lower affinity interactions that were missed by PBM and HT-SELEX. Nucleotides flanking high affinity binding sites create overlapping lower affinity sites, and the overlapping binding sites model provides a new framework for interpreting how non-coding variation influences transcriptional regulation and human disease.
The traditional model of how a TF finds its target DNA binding sites involves a combination of 3D hops and 1D sliding. As Shubham described, our results suggest that the intrinsic property of high-affinity sites to have overlapping lower-affinity sites confers them with a previously unrecognized mechanism by which they provide an energetic sink for TF binding. The PADIT-seq technology should be readily accessible to other labs. The assays are performed in 96-well plates using standard lab equipment, from which we then prepare sequencing libraries for Illumina sequencing. All code for analysis of the resulting PADIT-seq data are available on our lab GitHub page. There are numerous applications in which we aim to use PADIT-seq. As one example, identifying potential differences in the DNA binding activities of paralogous TFs can offer insights on differences in their regulatory functions and the mechanisms by which they are achieved. As another example, there are tens of thousands of TF coding variants whose effects on DNA binding activity are unknown. Coding mutations that alter the protein sequence of TFs have the potential to disrupt the expression of many hundreds or thousands of their target genes. Understanding the effects of such coding variants is part of the broader variant to function challenge in genomics. These are just 2 examples, but there are many more potential applications, including further development of the PADIT-seq technology that we aim to pursue.